on the HPRC clusters. I'm Wesley Bashir. I am with the HPRC enablement team, as well as with a bioinformatician with TIGS, or the Texas Institute for Genome Sciences and Society. That's headed by David Threadgill. I'm actually covering today for Dr. Michael Dickens, who's supposed to teach this course. He was unable to be with us in person, but he is going to be on the Zoom call. So if there are any questions that I can't handle, then we can go there and he should be able to provide an answer. I want today to be as interactive as possible. So if I'm covering something and I'm going too fast, or if you have questions about something, please speak up, both in class and ask questions in the chat on Zoom. So with that, I guess we should get started. All right, so we are going to be having some interactive exercises today. So if you have a account with HPRC and you want to do the exercises, go ahead and log in to Grace. If you don't know how to do that, don't worry, we'll cover that in a minute. There is going to be a short break, about 10 minutes, that's going to be halfway through this course. So don't worry about that. I'm going to be covering a variety of topics today. So I am going to go over the different sequencing platforms, the different technologies. So Illumina, Oxford Nanopore, PacBio, just a brief overview of some of those things. Some of the tools that are available on Grace to analyze these data. I'm going to go over quality control of short read Illumina data. So both evaluating the quality of the sequencing run, trimming the libraries, and we'll also look at some data normalization techniques and when you may need to use those. We'll also be looking at template job scripts. So these are scripts that are available for Grace users to run computationally intensive jobs on the Grace compute nodes. So if you say you want to map some reads to a particular genome, there are scripts available to help you accomplish that through our job submission process on the cluster. We also going to be analyzing some BAM alignment files. So we'll be doing that with both in the terminal and then on the portal with IGV and SAM Tools TView. We'll be briefly covering sequence variant calling, some of the tools there, as well as tools and a brief overview of RNA-seq and ShipSeq. And then we're going to end by looking at biocontainers. So there are a variety of ways to access the Grace cluster. The first, I guess, would be the HPRC portal. So this is web browser based, and all the HPRC software tools are available either as GUI or via the Unix command line. You can access the command line on the HPRC portal, which I'll show you how to do in a minute. But the HPRC portal is really great for using GUI apps. So like RStudio or IGV, which is a genome browser. IGV uses a lot of memory. So whenever you're loading a lot of alignment files, it's really nice to have that Grace backend to help with the memory. And even RStudio, I've had some projects that, so like differential expression, if you have a large number of samples, that can generate some really large matrices that can't really be handled by a desktop or a laptop. And so it's nice to have Grace where you can have a lot of memory to help load those and process all that information. You can also access Grace through the Unix command line. You do need to know Unix and Slurm, our job submission system. We offer primers on those, but those are typically at the very beginning of the semester. I know we offered one on Slurm. Did we offer one on Unix, Chris, do you know? Did we offer a primer on the command line? The short course? Okay. All right, great. Yeah, so I'd really recommend that. There's also like command line courses available through more traditional means. So I know Chris Blazier is a teacher that covers command line, as well as several other bioinformatics classes. But in order to do this, you need an SSH client on your computer. So if you're using a Mac, you can just use terminal. Windows, you can use something like Mobex Term, or again, you can access the via the HPRC portal. The last is the HPRC Maroon Galaxy. So this is web browser based, and Dr. Michael Dickens will be covering another short course focusing on using Galaxy. So this is kind of an interface between the user and a lot of tools that can be run via the command line, but it puts it in a nice interface that makes it a little bit, it may make it easier for some people to organize and run data. In order to use Maroon Galaxy, you need to apply first for an HPRC account, which hopefully you have, and then you need to request an additional Galaxy account. If the tool is not available that you want on Galaxy, you can always request HPRC to add one, or create a custom tool. Okay. So here's a quick example of how to use Mobex Term to log into the Grace portal. So first, you'll want to click here on session to begin it. And then right here in remote host, you'll type in grace.hprc.tamu.edu and put in your net ID. The great thing about Mobex Term is that it is X11 is enabled by default. X11 essentially allows you to view images whenever you're in the terminal. So it's a really nice tool. If you're going to SSH in, say via terminal on Mac, then you'll need to add the -X in order to enable X11. So this is the way that you connect with the HPRC portal on or through Grace using the HPRC portal. So I'm going to go ahead and try that. So up here where it says clusters, Grace Shell access, this will take you to a screen that looks like this. All right, so this is what pops up when you first log into Grace. Is that good? Okay. So after you log in, you see this message printed out. It's information on how to contact or the website for HPRC and some documentation and the YouTube channel, policy information regarding Grace usage. And this right here is kind of the message of the day, and that can change and give you important announcements about something like cluster maintenance or something like that. So make sure to check that. And then it prints out your disk quotas and usage. So right now in my Scratch directory, which is where you do all the heavy lifting, I'm using 8.4 terabytes out of a 15 terabyte limit. So that's really important to monitor. So right now, for example, I'm doing a project that's got several hundred samples in it, and aligning, there we go. All of those creates very large alignment files, right? And so, and would take, in the normal non-binary alignment file, that takes up quite a bit of room. So it's important to monitor that so you don't go over your quota. So, all right, so just to give you guys a brief overview of the anatomy of the cluster, just in case you are unaware. So whenever you access Grace, what you're doing is you're coming into a login node. And so that's what you're typing in. And these are for really simple jobs. So like things like file manipulation and job script preparation. And if you are going to run anything on the login node, it needs to be run in under an hour and use only a max of 8 cores. And you'll also need to watch your memory usage on that. Typically what you're doing is you're logging in here in order to submit a job that will be run on one of the compute nodes, which have a lot more resources. So these computational jobs can use up to 48 cores and or 360 gigabytes of memory. For the larger memory nodes, it can go up to 3 tera per compute node. And so these are for your bigger jobs, which typically is what we're going to be doing with NGS data. These jobs are going to take quite a bit longer than 60 minutes to run. So now I'm going to briefly go over some of the NGS technologies. First, I'm going to go over Illumina. And again, if you guys have any questions about these technologies and if it's something I don't cover, please feel free to ask. So Illumina is probably the most popular or most widely used sequencing technology right now. And it's short read data, so up to a max of 300 base pair read lengths. And there are a number of different Illumina machines that can be used. So they differ in their output as far as how much data they can generate, as well as the library configuration. So for example, the iSeq 100 is, I think, the smallest Illumina machine, and it can produce 4 to 8 million reads, whereas the NovaSeq 6000 is the largest, and it can produce, I think, 16 to 20 billion reads. So quite the range there, right? They do differ somewhat in what you can run, like the library configuration. So for example, the MySeq series can handle up to 300 base pair read lengths, which is really nice. So it can do the 2 by 300, which means paired in reads. Each of those reads is 300 base pairs. The NovaSeq and the HiSeq can do up to 250 base pair read length, I believe, and that's also quite nice. The rest, I think, can handle a max of 150 base pair reads. So, and all of these can handle the paired end, so not just single end reads. Illumina sequencing is highly accurate, so greater than 99% accuracy for the reads. So that makes it a really popular choice. You do run into some difficulties with it being, or limitations, or things that you need to overcome because they're short reads, but it's a great technology. So how these work, or how the libraries are generated. So typically you isolate whole genomic DNA, but what you'll need to do after you do that is fragment it in some way. So you can do this enzymatically or mechanically. And typically what you do is you shear the DNA down to 200 to 600 base pair fragments. Then you're going to ligate your adapters so that those fragments can attach to the Illumina flow cell, and then sequencing primers can find where to start the sequencing. And what that looks like for single in runs with these base pair fragments, this green portion here is what you'd be sequencing. So even though you have this 400 base pair fragment, you're only getting what here may be 100 base pairs. For paired end sequencing, which is the most common whenever you're dealing with either genomic data or full mRNA data. You'll use paired end. Single end is for, it's cheaper, but it's also used for things like small RNA, where having two reads that completely overlap doesn't really help you, right? So when really you're just interested in nucleotide fragments or RNA fragments that are smaller than 50 base pairs, a single end run is perfect for that. But for paired end runs, again, for things like genomic DNA and full-length mRNAs, this is what the fragment will look like. So you'll have this 400 base pair fragment, but you're going to be sequencing the first 100 or so base pairs from this end, first 100 or so base pairs from this end. You can also generate mate pair libraries. Are you guys familiar with these? No? Okay, so these are especially helpful for things like de novo assembly, whenever you're using only short read data. So essentially, whenever you have short read data, the paired end reads, they can build what are called contigs. So these are sequences that are thousands of base pairs long, but are they're contiguous in sequence. And then you can use mate pair libraries, which are further apart in origin on the genome, in order to scaffold these together. So you can stitch together your contigs to make longer sequences that should align with the whole chromosome. Does that make sense? Okay. So for these libraries, what you do is you take longer DNA fragments. So this example is 2,000 base pairs, but you can do 2,000, 5,000, 10,000, 15,000 base pair fragments. And it adds, it tagments the ends of these fragments here. It circularizes them, right? So that the ends that are actually, thousands of base pairs apart in the genome are now closely linked together. And then you fragment that circle and sequence only the regions that have this, this junction adapter inside of it. So again, you end up with something that looks like a 400 base pair fragment size, but those, what it's actually sequencing, the origin of those in the genome is several thousand base pairs apart. So again, here's the typical anatomy of double-stranded DNA and what you'd be sequencing with a paired end run. So again, you have like this read one of the pair, 100 base pair read length, read two, and then you have this small bit here that doesn't get sequenced, but overall the 400 base pair fragment size. And so knowing your targeted fragment size is important. Some programs request that information and use it to build like de Bruijn graphs or do de novo assemblies or things like mapping. So this is FASTQ format. This is what the reads look like in the text file after they're done sequencing. Have you guys experienced or having experience with FASTQs? Okay, so each read within the FASTQ file is made up of 4 lines, and there is a pattern. So first, we'll start each read with the @ symbol here, and then it has the read name. And so here is the read name, and then this indicates that it's the first in the pair. Down here, same read name, but it's second in the pair, right? And so that helps us keep track of the pairs of files as we go through and align them or do a de novo assembly. Next is the actual nucleotide sequence. So 100 base pairs here, followed by a plus always starts off this third line. Sometimes the read name is here as well. In this one, it is not. And then below that are the quality scores. All right, and these represent Fred-based scores. And so, for example, I think F is a quality score of 37 or something like that. And so the way these scores work, a quality score of 30 means the probability of it being incorrect or an error is 1 in 1,000. a quality score of 40, it has an error rate of 1 in 10,000. All right. So any questions on that? So whenever you generate your DNA or fragment your DNA to get it ready to be sequenced, you are targeting a specific link, but there is going to be some variation. So even though your DNA fragment links will be different, the sequence links should all be the same. So the left read 1 and right read 2, those sequences should all be 100 base pairs. What's going to differ is how far apart they are in the genome. So sometimes they're a little bit further apart. Sometimes they may even overlap. The MiSeq can actually perform the initial QC trimming before you're given the data. So in this case, you're going to have differences in read lengths between your left reads or your forward reads and your reverse reads. Typically in MiSeq or any sequencing that you do with Illumina that's on the longer side, so 150 and above, you're going to start to see a major drop in quality on the reverse reads. So those are going to be much shorter, and that's completely normal. This also depends what you're going to see whenever you receive your essentially raw Illumina data, is how it was demultiplexed coming off the machine, or how it was treated coming off the machine. So some labs will do adapter trimming and things like that before they even send you the data. Some labs just send you raw data. So that's something you need to check before you do anything downstream like mapping, is you're going to want to do a QC, look at the reads and see, how's the quality? Do we need to trim any adapters? Just how are things looking? So it's important. And there's quite a bit of variation between labs on what they do. And even within labs, sometimes data just gets handled differently. So another really interesting, relatively new sequencing technology is the Oxford Nanopore. Have you guys had any experience with this or done any nanopore sequencing? So this is actually, I love this bit of technology. It's really cool. They range in different sizes. So the Flongle is the smallest, and then they'll go up to the prometheon, or however you pronounce that. But essentially, all this is doing is taking which is essentially untreated DNA, and you put it directly on the flow cell, and then you can run sequencing like on your laptop. So it's really great, and it's got a lot of uses. So if, say, like you're in the field, you can be, away from, all sense of technology, and be able to sequence on site and have data generated that you can analyze there. So as you can imagine, it's got great applications for, ecology or disease outbreaks and things like that. Essentially, how it works is on the flow cell, there are these tiny pores that have these proteins on them. And this enzyme up here, as the DNA comes in, it's dragged through the pore and it's unwound by this enzyme. And right here on the membrane, there's an electrode for each pore connected to a sensor. And as the base pairs are pulled through the pores, it changes the current in a predictable way based on which nucleotide it is, and if there's any modifications of that nucleotide, say like methylation or something. So you can get methylation data and then sequence data. And another really cool thing about nanopore sequencing is that the data is generated as you're sequencing. So you don't need to go in and process the run to generate the FASTQ files. Those are being generated automatically. And so I think with Nanopore, it's every 10,000 or so reads that it gets, it writes those to a FASTQ file and keeps on sequencing. And so these runs, I think it depends on the machine, but they're typically run for like a day or so, but you're able to pull off data within a couple hours. You can start getting your data and looking at it. the one draw of nanopore technology is the error rate. So it is, right now, they're claiming it's greater than 95% accuracy. I haven't seen that yet. I'm still seeing around 91 to 93% accuracy. So depending on your application, you may want to go with a different technology, right? But if you're doing something, especially like smaller genomes, so we do this a lot for like bacterial genomes or something like that, or viral genomes, we can generate enough reads to get a nice consensus sequence that is, greater than 99% accuracy. So once you get enough coverage, the issues, it shouldn't be as big of a problem. But if you're doing something like sequencing a mammalian genome, or even like a bird genome, right, where you're getting 1 billion base pairs up to 3 or 4 billion base pairs, something like that, then that's becomes more of an issue with the error for this technology. So any questions on nanopore sequencing? It's really interesting, and it's fairly inexpensive, and it's if you guys have an experiment that you can maybe utilize this for, it's something that you can do in your lab on your laptop. So it's really kind of exciting.
So Nanopore would give methylation data without pre-treating the DNA with any sort of bisulfite.
I believe so. I don't. I believe that is the case. So lastly, PacBio long read sequencing. So this is the SQL sequencer. These things are massive. This is another really popular technology. So it does the long reads like Nanopore. It doesn't do quite as long as Nanopore. So Nanopore is claiming right now that they've seen reads up to, I think, 2 million base pairs in length. The reads from PacBio are typically shorter than that, but tens of thousands of base pairs is still a lot longer than your Illumina data. And there are different, the chemistry for PacBio has evolved quite a bit since it initially came on the market. And so now there are ways to get PacBio data that's almost just as accurate as Illumina data. So this is really nice. Some of the genome assemblies, for example, that are coming from the new PacBio chemistry are incredibly accurate and really clean. So I'll talk a bit more about this later on. But in the past, whenever we had only Illumina data and maybe the PacBio data wasn't as accurate, you'd have to do things like hybrid assemblies where you combine different technologies to produce full-length genome assemblies. And there were tools and software suites and pipelines to do that. Now, a lot of people are moving towards PacBio only assemblies, and they're getting full chromosome length assemblies quite easily. So it's really improved in the genomes that we're seeing now that we're getting relatively inexpensively are very high quality genomes, much better than even 4 or 5 years ago. So the way PacBio sequencing works, essentially we have this DNA fragment here, and this is what it's called a smart bell template right here. And essentially we're reading this right here, looping around and reading again, then looping around and reading again, and then looping around, right? So essentially you're producing this polymerase read that's got these positive strand sequences, and then the purple is the negative strand. And these are what are called subreads, right? And so even though your accuracy here, like within individual subreads, may be lower, once you combine all those subreads, you can call a consensus sequence and get these highly accurate circular consensus sequence, or those reads. So, so the accuracy then is somewhat dependent on the fragment length that you're sequencing. So if you have a shorter fragment like 5 kb, that's going to allow for more subreads, which increases your accuracy. The longer reads are going to tend to have fewer subreads, or excuse me, the longer fragments are going to tend to have fewer subreads, so they're going to be a little bit less accurate. But like I said, the chemistry has improved, and so now you can actually get decently long reads with very high accuracy. These are the Hifi reads that you can get from PacBio. These aren't really subreads. It's essentially the same technology, but they don't produce subreads. They produce the consensus sequence. So with PacBio now, you're not necessarily losing much accuracy, but you're gaining quite a bit of read length. So as compared to something like Illumina short read data, where you have those really small but highly accurate fragments. All right, any questions on PacBio? Okay. There are a number of PacBio sequencing tools available. So if you're going to take your sequence and just align it to a genome, an existing genome, Minimap2 is a great tool for this. It's also a great tool to use for Oxford Nanopore data. For genome assembly, Canoe is the tool that's put out there by PacBio. And so it's what a lot of people are using to produce genomes. We should note that the grid mode for Canoe is not supported on the HPRC clusters because of our accounting system right now, but we can still do Canoe assemblies on Grace. Unicycler is a good tool for the smaller bacterial genomes. WTDBG2 is much faster assembler than Canoe. It's very close, but it's not quite as complete as Canoe. Fly is another fantastic assembler, and it can take raw or corrected PacBio or Oxford Nanopore reads, and it also has the ability to do metagenomes, right? So you can imagine that's a much more difficult assembly process because with metagenomes, you're not going to have the same coverage for each this element that's present within the DNA, right? So Fly is good at parsing all that data out and doing decent assemblies for everything that's present within the sample. If you want to improve draft assemblies, so this is typically done with the PacBio data, or you can use your Illumina data to correct or improve draft assemblies. You can use something like Arrow on the HPRC. Canoe also has this tendency to produce redundant contigs or haplotigs. So it's important to remember to run a tool that can remove redundant sequences within the genome. So I read a paper recently, and they were comparing assemblers, and I forget the actual genome length, but say it was actually 3 million base pairs long. The other assemblers like Fly were producing genome assembly lengths of about 3 million, whereas with increasing data, Canoe started to produce 3.5 million, 4 million length, base pair length assemblies. So it's important to remove those redundant contigs or haplotigs, which you can do with purge haplotigs. Circulator is another tool that's commonly used. It's used to circularize sequences. So some assemblers don't have the ability, if you're sequencing something like a plasmid or a circular bacteria with a circular genome, they don't have the ability to actually circularize while they're assembling. And so you end up with a sequence that's got the same sequence on the 5' end as the 3' end, but in a linear format. And so Circulator can come in and circularize that and remove and correct the regions that are overlapping. So SmartLink, this is a tool suite that is available through PacBio. It's open source software, and there are a number of tools in there for handling PacBio data, which is what it was designed for. But some of these tools can also be used for like Nanopore long read technology. So quite a bit of stuff available within that software suite. So as I mentioned earlier, a common practice is whenever you're doing a new de novo assembly is to employ both long read technology and short read technology. That way you get the contiguity of assembly that the long reads provide with the accuracy of the Illumina short read data. And there are several hybrid assemblers available on the HPRC or the systems to do this. So Spades is a very popular one. It actually takes the PacBio subreads directly as input files. So there's no need to correct the subreads with the Illumina data prior to assembly. And it uses those long reads to close gaps within the scaffolds and to resolve difficult to sequence repetitive units. So things that are highly repetitive are difficult for short reads to resolve, right? So if you have a repetitive sequence that's, 1 KB or something like that, 2 KB, and you get a read from the middle of that sequence, if it happens within the genome, 100, 200, 1,000 times, that repetitive unit, that short read, the assembler can't tell which unit that actually belongs to, right? Because it's the same from each repetitive element. So that's what helps, the long reads help resolve those as well, because you can get actually unique sequence flanking the repetitive region to help you anchor those in position correctly in the genome. Mazurka is another tool for hybrid assembly. For this, it doesn't take the subreads, but you'll need all the long reads in a single FASTA file. Unicycler is an assembly pipeline for bacterial genomics. And this assembler actually has the capability to circularize without the need for a separate tool to do this. So there are a number of tools available on Grace. So where to find these? There are different ways to do this. So first, let's try this link here. Michael has this wiki page set up with a lot of the tools that are available on Grace and Terra, and they're organized here by essentially what you want to do. So do you need to do data normalization or clustering? There's the PacBio tools, the Nanopore tools, genome assembly. So you can click here. We can see we have Spades, Mazurka, like we talked about. There's other tools too. So things like all paths and SOAP de novo. Those are Illumina only assemblers that are very popular. And then the tools for scaffolding, gap filling, merging different assemblies. So these are how all these are organized. And so this is a really helpful tool. And then actually, let's do this. If you go and let's look at Canoe. This gives you additional information and provides job scripts for submitting these, which I'll cover in a little bit. So a really handy site. You can also access things through the terminal. So let me go ahead and log in here. Do you guys use Grace, or have you used it? Okay. How do you guys access it? Via terminal like this, or? Okay, not very talkative. All right. All right. So if you want to look up, say you know you have a certain tool that you're interested in using. So what are some things that you guys are interested in doing? Genome assembly, mapping? Is that a Illumina data? Okay, so a common mapping tool is Bowtie. So we can type in Bowtie 2 and it pulls up all of these different versions of Bowtie 2 that are available on Grace. There are a lot of things that are available on Grace that may not appear in the wiki. So make sure you check. You can check this way and check and see if it's there. If you don't know what tool you want to use, but you know what you want to do, so say you want to do assembly, you can type in module key assembly, and that searches the tool's descriptions and pulls up all the tools that are that have that word in the description. So that's really handy. But you can see it pulls up everything with assembly in the tool. So Buzzco right here is just a tool for assessing the quality of your genome after you finished assembling it. All right. So some tools are not available, or you can't show up with the module command. So Chime 2, that's available with Anaconda and won't come up, but it is there. So if you are unable to find the tool that you want to use and you want to install on Grace, you can just send an email, and Michael can typically have that installed fairly quickly. He's really responsive. He's a great team member at HPRC. So here's our first exercise. So if you guys want to do this, let's see. Log into Grace and we can just play around with this. All right, so the first one, let's look at, like I showed you, module spider gives you information. So let's type this one, module spider Bowtie 2. All right. All right, so there's several versions available for this. So now what we need to do is run module spider again, but this time we need to provide the specific package that we're available in. All right, and this provides the information we need to actually load this. So this is showing, all right, we need to load this module first before we can actually use Bowtie. All right, so let's go ahead and load both of those modules. Oh. All right, are we all there? Yes. I'm sorry, I can't hear you. I'm sorry, I'm having a hard time understanding. You can't log in on Grace. Do you have an account? You don't have a HPRC? Okay. Yeah, you'll need to apply for one of those first. So if you just want to follow along here, and then you can apply for one of those and be able to do the exercise. Okay. All right. Any other issues? Are we all here? Good. Okay. So let's go ahead now. We can look and see what we have loaded. All right. So this is showing all the modules now that we have loaded. You can see the GCC core 10.2 and Bowtie are loaded. And now to see which other modules we have that are compatible with the loaded ones, we can use the module avail command. So this will pull up the versions of SAM tools. Is what we're going to look for that are compatible with what we have right now. All right, so SAM tools 1.11. So let's load that. All right, great. And let's check again what we have. All right, and so now it's showing here SAM tools is available. All right, and then to clear all your modules, we do module purge. All right, and we'll run module list again to see what's there. And there it says no modules loaded. All right, so any questions about this? Good, pretty straightforward. So there are different tool chains available on Grace. These load different GCC compilers and or different MPIs and combinations of those. I think this one right here, the 2019B, FOSS 2019B, is where a lot of the bioinformatics tools, it's the tool chain those use. So for this, loading FOSS/2019B, that's also going to load GCC 8.3.0 and OpenMPI 3.1.4. And then it shows over here which version of Python is available within those tool chains. And to get a list of tool chains, you'll just need to type this in the terminal. So everything in the green blocks today is something that you can type in the terminal. So it's important to use a temporary directory when feasible. So some of the programs that you'll run as a bioinformatician or as a researcher, they'll generate a lot of temporary data, temporary files. So for example, if you're going to do an RNA seq de novo assembly, you'll probably run Trinity. And Trinity is going to generate thousands and thousands of intermediate files that it'll delete at the end of the assembly process. Similarly, I think if you're going to like annotate a genome and you use Maker, it produces a lot of files. And so you do have a file limit quota on your Grace account. So if you're going to do one of these things that writes this, you may actually exceed your file limit and it'll cause an issue with the program. So to get around that, we can use Tempter, which creates this temporary directory for writing all those temporary files. It is automatically assigned to each job, and it uses the disk space on the compute node and not in your Scratch folder. So it's not going to count against your file quota. There are some things you need to watch out for though. So if you're using some programs, you can run, and if an error pops up, you can address the error and then start the program from where it left off. So if it requires that it has those temporary files to restart from where it cut off, then you shouldn't use Tempter because those will be deleted after the error. Let's see. Yeah, and so it can also do things like speed up whatever job that you're running. And so to do that, so right here, Michael has this command typed out, doing a FASTQ to SAM file conversion, and here he assigns the Tempter to Tempter, right? So $Tempter, and that's how you utilize that function. So any questions on that? All right, so I'm going to go over now is quality control, but specifically for Illumina short read data. And so a popular tool to use for this is a tool called FastQC. And so what this does is it takes a subset of your library, approximately 200,000 reads, that you give it as your input as a FastQ file, and it assesses the quality of those reads at each position and produces a number of different graphs to help you visualize the quality of your data. One thing to know about running FastQC is that it will process using only one CPU core per file that you give it. So say you have 10 FastQs, or 10 FastQ files that you want to assess the quality on, and you only assign or use 4 nodes, it's going to take the first 4 files and start working on those. It'll queue the other 6. And then when one of those 4 files is done running, it'll pick up another one and process those as each one's completed until all the files have been processed. If you only have one FastQ file that you need to assess the quality of, then using 10 cores doesn't speed up the process, right? So it's a good idea to know exactly what's happening before you're submitting the job and requesting 10 cores when in reality only one would be used. And I'm going to talk, we'll talk and go over QC for Illumina data more extensively coming up. Digital normalization is something that you may need to do. So if you're doing a de novo assembly, either a genome de novo assembly or an RNA or transcriptome de novo assembly, it is possible to actually have too much data. So especially if you're sequencing something small like a bacterial genome, you can easily hit, 500x coverage, depending on what you're sequencing it on, like what platform. And this causes a number of issues. So first, it's going to increase the amount of memory that it takes to do the assembly. And it can also introduce errors into your assembly. So if you have, say, like 500x coverage of a genome, there's a chance that there's an error in the sequencing reads that it may pop up frequently enough that the assembly thinks it's true and no longer an error. Right. So by increasing that coverage, you're actually increasing the probability that it sees an error and considers it a part of the genome. So if you're, this is something that you'll want to do just for like de novo assemblies. It's not for other things like variant calling or like differential expression with RNA-seq data. You don't need to do normalization. Trinity, which is the most common transcriptome assembly software, it automatically normalizes reads to a depth of 50x. It uses a modified version of the seq-tk tool. Another tool to normalize reads would be the bbnorm script available in bbmap, which is on our clusters. All right, so now we're going to go over template job scripts. So Michael has generated a number of scripts for different tools so that it makes it easier. So for example, say I want to run software package that I've never run before. And so I'm still learning this, what I want to do. But I can go and look for one of Dr. Dickens' templates, and it actually shows you exactly, what you need to do to run it on one of our clusters. So let's go ahead and go look. Right, so FastQC, which we just talked about, it's got these GCAT templates available. So if we click here, all right, and so we'll go over this in a minute, but essentially this is the script that you'll need to submit from the login node to get the job to run on one of the compute nodes. And these are available for a number of different software packages. So it's really handy. It's a great resource. All right, so if you do go here to the GitHub and you want to just copy this directly from the web browser and put it in a text file for yourself, you'll want to remember to click on this raw first in order to do that. All right, this is another, the next exercise that we're going to do together. So what we're going to do is actually generate a GCAT template on in our Scratch folder. So let's do that. So first, we're going to make a directory on our Scratch. So you can just type in $Scratch, and that'll take you, that's the path to your Scratch folder, NGS class. Okay, so that made the directory. Now we're going to change into that directory. So whenever you're typing out these paths, it's helpful to hit tab, and that gives you your the full path spelled out. So we'll go into that directory now. All right, and then we'll run GCAT templates. All right, and what we want to do, let's expand this. So what we're interested in doing is running QC checks right now on Illumina data. So let's see what key.
I tried creating this branch directory, it says permission denied.
Okay, yes. So we have an issue with permissions being denied to create a Scratch directory. All right, it's.
I mean, when I first tried logging onto the base portal, it did say that could not find a Scratch. No such file or directory. Yeah.
When you logged onto let me see. So Community doesn't actually. Marinus, it doesn't look like he actually has a Scratch directory. So.
Oh, did you send email yesterday? Did you send email yesterday about this? We can resolve it offline.
Okay. All right. Yeah. So apologies, but we'll have to just follow along, and then that is weird. All right. So we're interested in doing QC, and so right here in this category, we'll hit 2. All right. QC. So we're gonna do fast Q quality evaluation. 1. All right, and we're gonna use fast QC. Let's use this tool. We'll just do it with the defaults. All right, and do we want to copy the script to our current directory? Y for yes. All right, so now let's list the contents of our current directory. All right, great. So by running that, we've generated the script, which will allow us to submit a job to the compute node. All right. So this is what, again, that file looks like. All right, so this top portion here, these are all the parameters that are read by the job scheduler. So right here, we're saying, do not export the current environment to the job. Right here, this is the job name. So you can change this to, whatever you think is meaningful. That way, if you need to monitor your job, you can type in SQ in your username. You can see which jobs you have running. So let me just do that real quick. So we run this command to see what jobs we have running. So right now I've got a job running on Crace mapping RNA-seq data. And so that name right here is what you see in the name file up here. All right, and then these right here. So this is an important one. This is the number of threads that you want to use. So we're asking for only 2 threads or 2 CPUs per task and 4 gigabytes of memory. This right here is where all your standard out will be written. And this right here, all of your standard error. This %J is going to be like the job ID number. What I actually find useful to do on these templates is to start off with that ID number. So it'll be %J dot standard out and %J dot standard error. And that way it'll know to open with a text editor every time. And so you don't have to go in and choose which tool to use to open up the file if it's ending, a file that's ending in a number. This right here is loading the modules. So module load fastqc. This right here is just a section of comments. Right, so everything with the hashtag or pound symbol, the computer isn't reading. This is where you're putting in our paired end information, so like the full paths to our fastq files. And this is assigning this slurm variable, right, slurm CPUs per task up here, all right, to a variable named threads. And then this right here, this line is the line that actually runs the command. So here we're calling fastqc -t this variable threads. So again, which is this slurm CPUs per task, which is 2. The output directory, right, which is right here. And we assign this essentially the current working directory to a variable called output directory. And then these 2 variables paired in 1 and paired in 2. Of note, I think this is a really good idea to do. Instead of typing in directly that we want to use 2 threads, oftentimes I'll go in and grab a template from something else I've done and modify it to do another project or to submit another job. And I've forgotten to change this up here and end up requesting something different down here. So this way you're always requesting what is expected by the job scheduler. And so this is a really good practice. All right, so let's run these. Actually, Yeah, let's run these. So this is when you'll need to have that X11 enabled, which I do believe I logged in with. Let me log it and log back in and make sure that I logged in correctly. All right, so having that X11 enabled allows me to pull this up, and I can actually edit it and save it back to my Scratch directory. Right, so this is really handy. This allows you to get around using things like VI, which is a text editor like inside the terminal. Which, have you guys used that before? I don't care for it. Some people like it. It's very confusing, right? Okay, not. So this is really great to be able to use this. And again, so it's the same file that we already went over, all the paired and stuff. All right, and now we're actually going to submit this job. So this is a really short job. It shouldn't take long. Let's go ahead and submit this, and then we'll come back to the results after a 10-minute break, okay? So to submit, we type in S batch. All right, requested service units of 2. That's awesome. All right, is everybody able to do this that's been following along? All right, let's take a 10-minute break. So we'll come back at 11:20, all right? I'm sorry, what? Mm-hmm.
What is it? I mean, I was searching for
You couldn't find it? Okay. So yeah, issue finding the GCAT templates. So actually, if you just here. This is the way I always go about getting those GCAT templates. Let's see, bioinformatics tools down here, and say I want to do, let's do read mapping.
I was looking for the EDI.
So
Yeah.
so right here. And then that'll take us here, see, and then we can click on one of these. And that takes us to the GitHub. All right, so yeah, let's take a break, and at 11:20 we'll pick back up. All right, so we got some people that are still out of the room, but it's past the 11:20 mark. So just to point out, I guess to those in the room and those that aren't aware of the chat, Michael said that you want to request about usually about 5 gigabytes of memory per FastQ file when you're running FastQC, and that sometimes that 2 gigabytes that we used isn't quite enough. Another person said, so if I have 10 FastQ files, I should ask for 10 CPUs and 5 gigs of RAM per sample. So you'd be requesting about 50 gigs total. All right. All right, so we've run that now. Now what we're going to do is unzip one of the results files and take a look at what we got back. Okay, so let's do that. So unzip. All right, great. And then we're going to run links to look at the HTML file, and then EOG to look at some of the images produced by FastQC. Nope. All right, and so you can see now this is just a basic output, and it's got all these different categories that it's looking at to assess for the quality. So you can see here, if you read down here, this shows you how to navigate using links. So up and down to move, and then right to follow it, and left to go back. So just looking at this, we passed the basic statistics, per base sequence quality, per tile sequence quality we passed. We'll go over these more coming up. What we did fail was the per base sequence content. So let's take a look at some of these images. We'll use. Oh, we also failed camera content. All right, so to look at this, we'll type in that command, EOG. All right, so that pulls up this file, and we can click here. Oh, yep, to look at other images that the FastQC produced. All right, so here is the quality score across all the bases. I'll go over that in more detail in a minute. That looks good. Sequence content across all bases. All right. And that is one who failed, right? So so right here, what you're looking at is essentially the percentage of each nucleotide making up the bases at that position. And you can see there's a lot of noise here, right? And what you typically want is these nice clean straight lines where there's an average distribution of the nucleotides across. all the samples or all the reads. So we'll look a bit more into that coming up. All right, so the first image that we looked at was the quality per nucleotide position in the reads. And so this is a kind of actually a very typical pattern of what you see in Illumina libraries, where it's, these are still good scores down here at the 5' end, but they creep up once you get, 20 or 30 nucleotides in, where you've got this really high quality of 40, right? So if you remember 40, that's an error rate of 1 in 10,000, right? That's fantastic quality. All right, so what we're looking at, so essentially for the subset of reads that FastQC looked at, it's taking these quality scores for each position and calculating like the average, right? So you've got the mean in red down here, this upper extreme, lower extreme, the lower quartile, upper quartile. So it's showing you the distribution of qualities at that position. And so it does this for individual nucleotides for the first 10, right? And then it starts to bin nucleotides together. So while these are for individual nucleotides positions 1 through 10, the next one would be 11 through 15, right? And that happens through the rest. So this right here, would cause us to take pause and look at our data and wonder what's happening. But this library was created using some sort of enzyme that fragments the DNA and allows you to ligate there. And so it's looking for a specific chimer, right, or sequence of nucleotides to where it can insert. And so that's why you get that bias right there. It's actually, they are part of the genome, so that is real, good quality reads, but you get this because of the way the library was created. So it's not something you want to trim off. It's actually part of the genome. Also, I think just to note, for trimming can have really major effects on mapping rates. So we'll talk a little bit more about that coming up. All right, and then same here, these cameras are popping up because of the way this library was created. All right. So this is one of the images produced by FastQC. This is quality of the actual flow cell. So the MySeq flow cell here, your blue is going to be high quality per tile, and when you get to the warmer colors, that's when the quality starts to drop off. And these are oriented in a weird way. So the actual top of the picture is the bottom of the flow cell, and the bottom picture is the top of the flow cell. So you can see here on the lower portion of this particular flow cell, the quality started to dip a little bit in these tiles. So some examples of libraries that have failed QC checks. This is a library that was made with an expired kit. So kit's been out for 9 months. They decided to run it anyway, and this is how the data came back. So this isn't really isn't usable. Yeah, I would just count my losses and move on in sequence with a different kit. But you can see here, like, some of these are ranging to, some of these values are below 20, which is the typical cutoff. So that's the low end that you want to keep, is anything, in the 20s. Here, the sequence prep adapters were still on the ends of the DNA library fragments. So that's what's causing this enrichment of these particular sequences. And so just need to trim this off, and then hopefully that will resolve the issue. All right, and this is an example of a faulty flow cell. So right here, again, this is the bottom of the flow cell, and you can see that it's really poor quality right here. And so I think this is something that Michael actually saw and sent the image to MySeq or to Illumina, and they said, yeah, that's just a faulty flow cell, and they sent them a new one to replace it and redo the run. So always good to check your data. All right, so like I said, there are, whenever you get your data back, it really varies on the lab, and sometimes within the lab on whether or not it's been trimmed, what the data looks like. So there are tools available to trim. So you can trim for both adapter content and for quality. Like I said, it does have some major implications for mapping rates and things like that, trimming. So for RNA-seq studies, if you're doing something like a differential expression study, you really want to keep your trimming light, because if you trim heavy and trim too much, then you can actually decrease your mapping rates because you'll get a larger number of non-unique alignments, right? So you may be tempted, if you see, something like this, right? You may be tempted to trim off, 20 base pairs, and you can do that clean. Like you trim 20 base pairs off the 5' end of every single read, and then you may want to trim 5 base pairs off the 3' end, where you get this really nice clean line. And yeah, it may look a little bit cleaner, but you're actually decreasing your mapping rates. So just something to keep in mind. And it's not proper bioinformatics if you haven't done it, more than once on the same sample. So always good to play around with parameters and things like that to get the best results. So there are a number of tools available for trimming. So this is one of the recommended tools, Trim-O-Matic. I like this one. I also like CutAdapt. That's a really good tool. These are recommended because they actually maintain proper read pairing. So some tools may trim out an entire read, like one of the pair, but still write the other read that passes to file. And so it ends up throwing off the files because one file may contain more reads than, say your forward reads may have more reads than your reverse, right? Because some of your reverse reads got completely trimmed out. That's going to throw a huge error for the assembler or for the mapper or something like that. So you want to make sure you're using a tool that maintains that proper pairing. And so Trim-O-Matic and CutAdapt both will. And so again, this trims reads based on the quality scores. And like I said, you can trim reads like the same number of bases from the 5' end for all reads, or you can also use it like a sliding window to calculate the average quality, the ends of sequences. You also need to decide what you want to do with reads with ends. Some assemblers replace ends with random bases, so this is going to obviously affect your results. Let's look at this. So for whenever you're running Trim-O-Matic, you'll need to provide the adapters. CutAdapt has the ability, it automatically detects which Illumina adapters were used in the library. So you can provide it with the adapter sequence, or you can let it auto detect. There we go. All right, so when we loaded this module, it says, it brings up this helpful hint on how to actually run this. So it says, you have to run Java jar and then give it this path before actually running the traumatic file. We're going to look just at the adapters. All right, so yeah, so these are already available on Grace, so you can just run, and if you used one of these, you can just give it that file path to trim those adapters. Yes.
Is it necessary to trim off the adapter and go through mapping? Because if I go through the raw data, so it won't have, so it will be the adapter as I won't.
So it will. It depends on how the program handles it, but it can actually interfere with the alignment, and because you have those adapters on there, it's going to assume that it may align like the actual proper portion of the read, but it still has to deal with those adapter sequences. So that's going to lower your mapping quality. It may force it to actually not align. And so that's not a good idea.
If I trim off too much data, too much adapter or too much sequencing, there will be like missing any information.
Not if you're trimming just adapters. Yeah, so if you just do like a default trimming, what's going to happen is it'll trim for those adapters and low quality sequences, and it'll retain everything else. So you're not actually losing any information because the adapter doesn't come from the genome, right? So yeah, it'll just preserve what is true, genomic or RNA sequence and map that. So. Sorry, somebody had asked a question about trying to align reads without trimming adapters first. So just a brief overview again to review this real quick. The anatomy of essentially what these reads look like is 400 base pair fragment size. Read 1 coming from this strand, read 2 from this, and then the FASTQ format with your read name, always with the @ symbol, the actual nucleotide sequence plus and the qualities for both pairs. So whenever you're trimming paired end short sequence reads, so Illumina reads, if you do a QC trim, a lot of times the read 2 is going to end up being much shorter. That's because read 2 is processed later in the sequencing run. So essentially it processes all of the read 1, right? And then it does that technology where it bends the DNA fragment over and sequences the reverse read that way. And so towards the end of that, because they're adding nucleotides to the flow cell, washing and doing all that, it starts to build up noise. And so the sequencing quality tends to go down towards the end of the run. And that's especially the case whenever you're doing the much longer read links too. So like a MySeq or like a 2x 250 or 2x300, something like that. And so on the whenever we're trimming, there is typically a minimum read length cutoff. So we'll say just get rid of any read that's less than a certain number. So the example is 40. So we trimmed read 1, read 2. So we got still maintained 100 bases here, 50 here, and then this would be the result of the trim files, right? But if you get below trimming, get below a certain threshold, then what should happen is the trimming program should write this to a single end file, right? So you end up with 3 separate files. So the ones corresponding to the reads that maintained pairs, so that are still properly paired, and then additional reads whose mate was lost. You may also need to merge overlapping paired end reads. So remember we talked about the fragment length. So say you shoot for 400, there's still going to be some variation. And some of those fragments may be so short that the reads actually overlap towards the end of each read. It's a good idea to merge those so that you end up with just one longer read. Because the read quality tends to drop off towards the end for both, merging can help kind of correct any errors that might be there and produce a higher quality, still useful longer contiguous sequence. And there are a number of tools available to do this. So Flash, BBMap, CopeRead, and Pair. All right, so let's talk about a little bit about mapping reads to reference assembly. Some very popular tools are BWA and Bowtie 2. Those are pretty much this. Well, no, I can't say that. There's BBMap's also fairly popular, but you can't really go wrong with BWA or Bowtie 2. There are several algorithms within BWA, so you'll need to do some reading and make sure you're using the correct one. For these aligners, they always need genome index files. So that process, essentially, it builds like a library of sequence information from the genome that helps it align reads quickly to where they actually originate from within the genome. And this process can take several hours to build an index. So there are some available. Let's go look. All right. So Dr. Dickens has pooled, you can get genomes from a variety of sources. So like Ensemble and NCBI are two common places to get genomes from. And they do have different attributes, right? So Ensemble, the genome annotations there are slightly different than the ones from NCBI, right? They still all contain roughly the same genes, but I think Ensemble does sometimes have more sequences or genes that may have more information about them there than NCBI, and maybe even vice versa. So it depends on the organism and what you want to use. So there are some very common genomes or species that are used. Let's see what's in the Ensemble one. All right, so actually in Ensemble, we have TAIR10. So that's the Arabidopsis genome. All right, and NCBI, we've got MM39, which is the mus musculus, and then MRAT B9BN7, which is the Rattus norvegicus. So it's just a good idea to look here if you before mapping, if your lab uses a commonly used organism, it sometimes can save you a lot of time because those resources are already there. So yeah, Arabidopsis potato. All right, so yeah, there's different ones available. All right, now we're going to go through some exercises on visualizing BAM alignment files. So let's do this. We're going to do this exercise. So make sure you're in your NGS class directory if you're able to set that up. And you're going to create symbolic links to the example files in your working directory. Do this for the alignment file there, the DR34 SAM, and the genome FASTA file. You don't need to do this when you're using your own data. This is just for demonstration purposes. So I'm going to do this as well. And if you have the PowerPoint, you can just actually copy and paste. Whenever you're typing long paths like this, it's always a good idea to use the tab to finish out. It's quicker and it ensures that you're getting the path correct. All right, has everybody been able to do that? We good? Okay. So whenever we have, whenever you first align with like BWA or star or bowtie 2, it produces a SAM file. It's essentially just a text file that you can read, and you can read it using a common just Linux command like more. So let's try that, more dr34.sam. All right, so you can see here it prints out this header and starts reading the file. And we have these alignments. All right. But really what we want to do is generate a compressed version of the SAM files that helps us save space and move files around much more quickly. So let's go ahead and load these. And we're going to sort and compress in the same step. So I'm going to do this. All right, and then whenever you're doing, if you want to visualize the data, or if you're going to be doing something like differential expression, where you're counting reads mapping to certain genes, you'll always need to sort the alignment file. And typically you want to sort that on coordinate position, so where the reads are mapping. So essentially you're grouping all the reads that are mapping to a particular portion of the genome in the file together. All right, so that makes it easier for like SAMtools TView to load the reads, or something like IGV to load the reads. So let's go ahead and do this. So SAMtools sort, this -@, that's the number of threads to use, right? -m, the amount of memory we want. -O, this is going to be the output, and then we're giving it the SAM file with which to do the command. So you'll also need to index your alignment file in order to open it in a viewer. All right, so now we can see we've got links to the SAM and the FASTA file there, the binary BAM file that we created, and its index. Everybody good? All right. So now that it's in binary format, we can't just type in more and read the file, right? It's not human readable anymore. So we'll need to use SAMtools view in order to do this. So let's just do SAMtools view. Right, and so what that does, we did SAMtools view. which will allows us to convert it to human readable format, and then we pipe that into more. And the command this way just produces the alignments, so it doesn't give you the header or any information like that. But you can go through the alignments this way. If you want to view only the header, you give the flag dash capital H. All right, so we didn't need more that time because it just popped out only the header, which is much smaller. And so you can see up here now that this is a coordinate sorted file, which is good. Then it gives you information on the sequences within the genome. So apparently there are 8 sequences within this genome, labeled 1 through 7, and then something labeled R. And it gives you the links of those different sequences over here. All right, and then it gives you, this right here is the information on how this file was generated, and additional information right there about, yeah, how the file was generated. All right, so to view the header and the alignments, you'll use the dash little h. I'll show you why you need that more. All right, so I'm just gonna type this in without doing the more first. And you can see that it's just looping through this, and it's too fast for us to read. So if this happens, just Control C, let's clear that. All right, and then more. All right, and now we can go through and actually look at the file. All right, everybody have a good handle on SAMTools view? All right. So let's just briefly go over kind of what the alignment file looks like. First, you have this read ID. Then you have a flag, which is made up of different bits. The sum of the bits. We'll go over that in a minute. They all have different meanings to give you information about how that read mapped, where it mapped, or what chromosome it mapped to, and then the actual coordinate. So let's go down here first. Flag of 99. This is a great tool you can use and explore more later. Just Google explain SAM flags, and this will come up, and you can click around and it'll produce SAM flags. So for 99, that's going to be a read that is paired, it's mapped in the proper pair, it is mate is on the reverse strand, and it's the first in pair. All right, so quite a bit of information just in the number 99. And we can also use these flags to filter. So for example, if you want to filter BAM Alliance and keep only reads that are mapped in the proper pair, you would type this command, right? Which gives you, it does both the reads and the header. This tells it it's in BAM format or output in BAM format. -F2, this is keep. Lowercase f means keep only reads that have this bit, and then the BAM, and then it writes it out to file. All right, if you want to keep all reads except, let's say, things that are PCR or optical duplicates, you can do that with this function right here, SAMTools view again, but this time we have a capital F, which means leave these out, ones with this bit. There are several tools available too to remove duplicates. Just a side note, that's something that's important to do, especially if you have a lot of PCR cycles or something that can really bias your data. But this is another way to do it. So any questions so far? Good. All right, let's look at, whenever you're done with the alignment, it's a good idea to look and see what actually aligned. A good tool for that is the SAMTools FlagStat. All right, and that's going to pop up essentially all the reads. So there are 150,000 reads that passed QC. So it looks like 93% of these mapped. However, only about 57% of these are properly paired. So that's not great. But this tool allows us to get that initial, essentially QC of the alignment, right? All right. So one way to view an alignment is using the Samuel's T-view function. All right. So let's try this. All right, so here are the reads, right? So these in blue are indicating or reads aligning. But you can see up here that it looks like it's the reference sequence here is only ends, and that's because we didn't give it a reference sequence. So let's try that first. Yeah. Use up. All right. So now we have, these are still the reads. Here's the actual reference sequence, and we can scroll through doing this. All right, so if you're kind of lost and don't know exactly how to do this, you can always type in a question mark, and that's going to produce this help. So you can see here there's a lot of different functions that increase usability. So if you want to move quickly, you can use this H function. Let's try that. Let's do L. See, now I'm cruising through real fast. Right, when you're going through millions of base pairs, that could be helpful. You can go to a specific position. Scroll less quickly. So just a good idea to have that. Remember that typing in question mark brings up that help menu. All right. So here, if we're looking here, if you type in this command, it'll take you to this particular position. So let's just go ahead and do this. All right. And so now what this is this looks like it's a homozygous mutation. So right, if we're working with a diploid organism, that means it's homozygous in that position for both. And what this means is that C is the reference sequence, but our sample contains only A's. The dots here mean that it matches the forward strand. The commas matches the reverse strand. Capital A, it doesn't match the forward strand, and lowercase, it doesn't match the reverse strand. All right, I do want to go over, so SamTools T-View is only one tool we can use to visualize alignments. I do want to cover, let's see here. So this is one great thing about accessing through the portal, is that you can come in and look at interactive apps. And what do we want to do? IGV. So you'll have to request to be able to log in and look. So we're just going to request 1 hour, one number of cores. We don't need that. We'll just keep it there. And you can put in the account in your email. So if there's a wait time, it may take a while for your session to launch. We'll just give this a go and see. Hopefully it works. So it's queued right now. you can, if you put your email in, you can walk away and it'll send an email whenever it's ready. Yesterday when I did this, it took about 30 seconds and I was able to log in. There we go, and it's running, right? So let's launch IGV. All right, so because I did this yesterday, it's cheating for you guys. So to load a genome, you want to go to load genome from file. This puts me in my, looks like home scratch. So, or my home folder, not my scratch folder. So we need to go to our scratch. Right, and then we can load that FASTA file. All right, so now it's showing all these chromosomes or all these sequences together. So you remember we had chromosomes 1 through 7, and then the last one named R. But we want to load our alignment file. All right, and that's the one. All right, and you can see that nothing's popping up, right? But we'll have to zoom in. There we go. All right, so this is a good way to visualize the data as well. So you can see it's right here where it's matching. It's gray. And where there are mismatches, it produces different colors. These blue reads indicate that there's something off about the fragment size that it was expecting based on the rest of the library. And so these reads are actually overlapping reads, it appears. But you can scroll in further, and then eventually you get down to nucleotide level. And then you can go anywhere you want, just typing in up here. So well, it's not allowing me to paste for some reason. But you can just essentially go there, and if you like, you generate a VCF file, we'll say, which is something we're about to go over. It's a variant call file, and it gives you, you've got this position here that's got a variant in these samples. So you can go directly to that and kind of inspect and make sure that the variant is real. Okay. So any questions on IGV or SAMTools TView? We're about to move on. Okay. So IGV can actually be run on your local machine too. You can download it and run it. All right, so we are going to go briefly over sequence variant calling and some of the tools that are available to do that. It typically starts, what you're going to do is align your reads to the genome, so using like bow tie or BWA. And then you can do things like you'll need to process the alignment, so you need to sort it like we did. You need to remove PCR duplicates. For certain pipelines, others like GATK don't really require QC trimming. But like I said, you can mark PCR duplicates to remove them. A common tool for that is Picard. Picard is a suite of tools that's commonly used for different manipulation, different ways to manipulate alignment files and things like that. And so there are, as you probably saw when we were looking in IGV, excuse me, there can be sequencing errors in your reads. And so what we need to do is differentiate between SNPs, like actual mutations and sequencing errors. So in order to do that, you definitely need high enough coverage. So typically, you need above 10x to do this. If you're going to be looking at indels or structural variants, you're going to need more reads than that, or higher coverage than that. And in order to actually, it depends on your data type. So for some data sets, like the mutation may only occur in some of the cells or something like that, you'll actually need it to be at a fairly high rate in order to recover it. That's not the case in case it's like like an actual mutation that was inherited, right? Then most of the reads should contain it. Strand bias may actually be a result of whatever sequencing platform you use, so you also need to take care of that. All right, and we'll go over variant call format here in a second. Oh, and we do need to be able to identify what are called multiple nucleotide polymorphisms. So MMPs, that'd be 2 SNPs within the single codon. So right here, if you have this mutation, it results in a change in the translation. Actually, if you have both, it puts in a stop codon. So more about PCR duplicates. Those are just artifacts resulting from the PCR amplification. And like I said, depending on how many cycles you did for the PCR, it may be a much larger problem. If you did, a lot of cycles, if you're starting with a low amount of DNA, and those should definitely be removed or marked so that you're not interfering with, the frequency of variants or gene expression levels. It's not actually something that's biological. It's just an artifact of PCR. All right. Here's some of the software tools that we have available to use. GATK is maintained by the Broad Institute, right, which is an institute that's partnered between, I think, Harvard and MIT, right? So it's a really world-class institute, and they are constantly maintaining this suite of programs. So GATK has it can do variant calling, and as well as some other things as well. So in this, there's no need to QC trim reads. They've got a best practices pipeline that you can follow to ensure that you're following what are standard procedures that will be readily accepted by whatever journal that you're trying to publish in or something like that. You may need a set of known SNPs from your species. So if you're working on something like human or mouse, that's really easy to get, or rat, or even Rabbitopsis, something that's common, those database SNPs are available. If you're working on something that's more novel, you'll have to generate one of these. Essentially what you do is you call your variants and then cycle through and bootstrap and get all the variants that are correct, and that generates kind of a known set of mutations or variants that can be used as kind of a way to examine the other variants that are found within the samples, and to make sure that those are high quality true SNPs or small indels. In order to use GATK, you'll definitely need to be able to use SAM tools and Picard. Other variant calling tools are like SAM tools and BCF tools has some function to call SNPs, Freebase and VerScan. So there's a number of options. GATK seems to be, at least in my opinion, kind of the current gold standard on which variant caller to use, but the other ones are good as well. So here's an example of the VCF file format. This is what prints out after you run one of these. This is cut off, so there are more samples here. So this actually has, I guess, 5 total samples. It's somewhat similar to a SAM file. You have this header here. Let's go over this. There's quite a bit of information here. So this right here is the info section, right, for all the samples combined. So for example, this right here is the total read depth, the allele frequency, and this is, you know, if it's a member of that the dbSNP database that you gave it. So this right here is the filter. Quality below 10 is Q10. S50 is less than 50% of the samples have the data. And right here is where you start to get into the individual sample information. So this is a format, so it goes genotype, genotype quality, and the depth, and this is haplotype quality. So right here, you can see this is the genotype. So 0, the pipe symbol 0, that indicates that these are phased, so they're found right on the same chromosome. And here is the genotype quality 48. If it's a / like this, indicates the variants are not phased, right? So and right, so 0 means it's the same as the reference, right? And then for here we have the 2 alternative SNPs, so 1 and 2. This person has this sample has, you know, 1 paternal or 1 reference sequence and 1 mutation. Any questions? All right, so this is a Venn diagram. So they essentially went in and called variants with each of these 3 tools. And so you can see there's quite a bit of overlap in the variants that were detected, but there is some differences. So like GATK picked up some variants that the other 2 didn't, and these share as well. Some people have called variants. I've seen they've used multiple tools to call variants, and then only use the variants that were found by all the tools is a way to like filter and make sure that you have high quality variant data. All right, so once you have the variant called, there are several tools available to look at the consequence of that variant. So Provian is one of those tools. And what it does is it looks at, it looks to see if that nucleotide change caused an amino acid change, and it looks at the severity of that by comparing across multiple species. So with this tool, if you have a score below or equal to -2.5, it's considered deleterious, right, or harmful. So if you look at these, they're frequently in positions. If you look at ones that have, you know, very low scores, they're typically in positions that are highly conserved across species, right? So that any change in there may have a considerable effect on the function of the protein. So yeah, Provian is one good tool for that. You can also annotate variants with SNPF. So what you need is a VCF file, a reference sequence with gene annotation. So that's going to be a GFF or a GTF file from Ensemble or NCBI. SNPF annotates the VCF. There are a number of large databases available to it, but you can also build your own. And it also annotates those multiple nucleotide polymorphisms. You can see right down here. So here's output from looking at alignment. And this is a diploid individual. So you can see here that this SNP is not occurring in all the reads. It looks like it's occurring in about half of them. So it's a heterozygous SNP, whereas this right here is a homozygous SNP. You know, all the reads have this single nucleotide variant going from was an A and now is a G. All right, so this is specific to the ion torrent. I actually haven't worked with the ion torrent data before, but it can have sequencing strand bias. So here in this alignment, the strands are color coded. So blue is the positive and red the negative strand. And you can see that all these mutations, right, are occurring in just the forward aligning reads, and so that shouldn't be the case based on probability. So you can actually there is a way to identify and estimate strand bias using values in the VCF. So if you ever use ion torrent data, just be wary of that. All right, so a brief overview of RNA-seq. We're going to be doing an RNA-seq short course later in the semester. And so in that, we'll briefly discuss kind of transcriptome assembly, but it's mainly going to be focused on differential expression. And we're actually going to be doing a full differential expression work through in R. We're going to start by mapping the reads and doing all that, and then going into R to calculate differential expression. So there are a number of programs available on Grace for this. HiSat2 and STAR are commonly used aligners. Top Hat is one that a lot of people still use. The author of Top Hat is actually the author of HiSat, and he begs people to stop using Top Hat. It's deprecated. It's old. It's not as efficient as HiSat, but it's still really commonly used. It gained a lot of popularity, and diehards have yet to migrate over to HiSat2. STAR is really good as well. I prefer HiSat2. It requires less memory and it appears to run faster. And I had better mapping results with HiSat2 than I have with STAR, but STAR is really good. Some packages for actually dealing with or looking at differential expression, DE-seq2 and EDGE-R, both are fine. Transcriptome assembly, you can use Trinity is the standard now. Oasis and SoapNovaTrans are older programs, but for reference-based, you can do Trinity or string tie. You can also do like cufflinks after an alignment to build your annotation. So you can do variant calling with RNA-seq. I haven't really seen much of that, but it is possible to do. You can also use RNA-seq to scaffold genome assemblies. All right, so this is something that pops up a lot at my my job over in TIGS is people doing differential expression studies and whether or not to spend money on getting deeper sequencing or more biological replicates. And the question is, what should we do? And you should always go for more biological replicates. That's going to increase your capability to accurately detect differentially expressed genes. So I read a study, and what you see a lot of people do is the bare minimum of 3 control and 3 treatment, right? And and that's fine. That's the minimum you can use. But the more biological replicates you have, you actually increase the number of differentially expressed genes that you can accurately detect. And so I believe this study recommended a minimum of 6 per group, and actually recommended doing 12 per group is where you start to recover all the differentially expressed genes that are accurately or actually differentially expressed between the groups.
Is there any requirement of, if you're going for differential expression, then recommended to go for paired and data or single?
I would recommend paired and data for like mRNA. Yeah, if you're doing either ribosomal depletion or poly-A selection, yeah, it's just better to use. Just get more data. But yeah, with short reads, it doesn't. somebody does paired end sequencing with short end reads, I just throw away the second read. So transcriptome assembly, this isn't, well, there are a number of ways to do this. So you can just type in, module spider Trinity or HiSet2 and cufflinks to get, more of an idea. Without a reference genome, Trinity is the one I would recommend. So how Trinity works, it's named Trinity because there's 3 stages. So essentially, Inchworm takes the raw reads and makes these linear contigs. Chrysalis essentially clusters these contigs, and Butterfly creates the actual final transcripts and isoforms. Ideally, you'd want one graph per gene or transcript. What you end up a lot with Trinity is you get hundreds of thousands of transcripts depending on species, and it's just an overestimate of what's actually there. So if you have the ability to do reference-based, then that's the way I'd recommend going. All right, so running Trinity on Grace, like I mentioned earlier, it's one of those ones that runs or that uses tons of intermediate files. So you will either need to get a quota increase for your account, or use the Tempter, use a temporary directory to write all those files to. Also, it's recommended not to run multiple Trinity jobs at once. Trinity is one of those programs that that can restart if there's an error. So if you're using temporary directory, you'll need to rsync the temporary directory to your Scratch in order to resume with those checkpoints, or from those checkpoints. And there are GCAT templates available for the Trinity. ChIP-seq, so chromatin immunoprecipitation. Has anybody done this or doing this? Right. So this is a really cool technique. Essentially, you're looking at protein DNA interactions. I've only got to do a handful of these, but they're really interesting when I get to do them. What you do is you cross-link, bind the proteins to the DNA, and then you isolate the chromatin and shear it and fragment it. And then you precipitate with a protein-specific antibody. And what that's going to do is enrich your DNA for for sequences that were bound to that particular protein, transcription factor, what have you. Add the adapters and you sequence and run. This is a view of an alignment of ChIP-seq reads back to the reference. So you can see we get this large peak here in the middle, and that's because we've enriched for reads binding FOXA3. And so those will appear, throughout the genome. And then you can go in and grab this flanking sequence, right, and look around and generate kind of a motif or consensus style to look at what actual DNA sequence it's binding to. So this is done with the R package seq logo. So a number of tools available. Max2, that's very common. Peak splitter, Peak Ranger, Broad Peak. For the different steps of the ChIP-seq, these are all available. And there's some really good pipelines available online to follow in order to analyze the ChIP-seq data. All right, so for interactive apps on the HPRC, you are charged service units for things like IGV and R. You're not charged for accessing the shell. If you want to run a GUI app that's not on the list, you can use the VNC app and launch the GUI from the VNC terminal. In order to access this, you must have, if you're off campus, the VPN running. So just a heads up. Briefly, just so you know that this is out here, I haven't actually used this, but if you're doing something that requires a large number of just single core jobs, like running a blast, you can use this TAMU launcher. Essentially, you have this command file with all of the commands that you need to run, and TAMU launcher will organize that and ensure that they all run. So if you have, like, say, thousands of blast jobs, those take, quite a while. So if there's any way to parallelize the process, then that's good. So looking at TAMU launcher, we are running out of time. And this is the last thing we're going to cover is biocontainers in 3 minutes. So just quickly, biocontainers are containers that have one or more software packages installed. It's a single image that contains, one or more software components. So you may have heard of Docker or Singularity. Those are used to build and access the biocontainer. The container does not have the entire OS, just like components needed to for running, particular software. Some pros, the software is already installed, and it's got all the correct versions together and all the correct dependencies, and it's just one file versus, Kana environment, which can have thousands of files. The latest software, though, is not always, available, and then software dependencies within a container may not be the version that you actually want to use. Docker is not available on the HPRC clusters, but Singularity is. So you have to convert those to Singularity image, anything from Docker. And it's only available on the compute nodes, not the login nodes. And the compute nodes don't actually have internet access, so you'll have to enable a proxy to use it. You can do this. So here's essentially a quick rundown. Connecting to a HPRC portal, srun for an interactive job, load the web proxy, then convert right here. So this essentially setting up these variables, and then Singularity to pull the Docker container that you want to use and run it. So I guess we'll leave it at that. If you have any questions about biocontainers, just reach out, and the help desk will be able to help you through it. All right, and lastly, just real quick, using that alias. So there are a lot of things that we do over and over again. Like I type in SQ-U, my name, to look at the jobs that are running. If you add like alias Q equals this to your bash RC file, every time you type Q into terminal and hit enter, it'll run this full command. So just a quick tip and trick from Michael. It's really handy. So you don't have to type in SQ-U, over and over again. All right, and then if you ever need any help, just send an email to help@hprc.tamu.edu, just regarding any bioinformatics tools, if you want something installed. But first, spend some time investigating the error. Like, we need to know the read log files and everything like that. And then if you do have an issue, include details about the problem, like the job ID, which cluster you were using, if it was on Galaxy, the software, modules, and all that. So with that, I'll take any questions. Yes.
Is there any information on the short course or RNA-seq?
I don't think those have been announced yet. The RNA-seq short course, it's on a Wednesday at the end of the month. It's Wednesday. So, okay, yeah, they'll be posted shortly. Okay, but yeah, it's towards the end of the month.